we are currently looking at working together with an ASTM collaboration group to answer your very question, because that is something that we have been approached in the standard committees, and this question has been arised by many, and we don't know the answer. So we have created this group that's going to do a round-robin testing and see if that's even applicable.
What if one conversation could save a life? What if one new process or new product could save a patient from surgical harm? Broadcasting from a hospital basement near you, you are listening to the Beyond Clean podcast, the global voice of sterile processing. Each week we spotlight the best practices, products and people from around the world who are disrupting our profession and helping you fight dirty. Every instrument, every time. Join us as we have the kinds of conversation that matter and give our industry the kind of voice that it deserves. Nyt on aika mennä puhdastakin puhtaammaksi. And now it's time to go beyond clean.
This week on Beyond Clean, we speak with Alpa Patel, principal scientist at Nelson Laboratories, LLC, and we're going to be talking about validation and the science behind validation with the chemicals, which piggybacks really well off our last episode where we talked about really chemicals used in bedside cleaning and how do we actually know that they're working. And so we're going to take a much deeper dive with Alpa today, Hank.
Yeah, I'm having flashbacks to my childhood, sitting in front of Bill Nye, the Science Guy. Bill Nye, the Science Guy. And I've been trying to think about like an alternative theme, for Alpa. But the words are not coming to me. But I know at least by the end of this episode, I'm going to have a theme song for the science discussion that we're about to have, really digging into. Obviously, we just talked to Chris Murphy last week about science, but we're getting into that deep science on the validation side for testing all this. Chris is talking about developing the chemicals themselves. Now we get to figure out, hey, are those chemicals that are created, do they actually work according to the IFUs and how those IFUs kind of mesh with everything that's been done? And then how do the users follow it appropriately? So those are the questions that I'm going to be bringing into today's conversation.
All right. Well, by the end, you will be saying to yourself, she blinded me with science. We're going to be right back after a short break with Alpa Patel. I'm Justin Poulin.
And I'm Hank Balch.
From 17 Studios, you're listening to Beyond Clean, the global voice of sterile processing.
Joining us now is Alpa Patel, principal scientist at Nelson Laboratories, LLC. And on the last episode of Beyond Clean, we talked a lot about chemicals and specifically the use in the GI space and related to bedside cleaning. But one of the questions came up was, how do we make sure that these chemicals are doing what we think they're doing in our own departments? It's kind of a validation versus verification. I think there was a big question mark about how end users and frontline workers could actually verify. But today we're going to cover the other half of that question, which is the verification piece. And so Alpa, we're really excited to have you on the show to continue this discussion in season 15 of Beyond Clean.
Thank you for having me.
So I'd like to start by just having you talk a little bit about how you found this industry. And I know we're going to be diving into the fact that you're a scientist and we're going to talk about validation and chemicals, but how did you find your way to your current role?
So luckily, this was my first job right out of school. When I moved to Utah from Canada, I was looking for a job and I am a microbiologist. So I was looking for a lab that I can go work for. And there it was Nelson Laboratories, a micro lab. And I applied for a position in there and I got the position and it was in healthcare reprocessing. At that time, I didn't know what healthcare reprocessing was. But when I started in there, it was very clear and very complicated at the same time that I'm working with so many different types of reusable medical devices. And that's how I started my career in the sterile processing industry.
All right. So you're very much on the other side of what happens in this industry. Many people who listen to the Beyond Clean podcast are out there working in healthcare organizations, hospitals and surgery centers and dental clinics, et cetera, and doing reprocessing, but they don't necessarily know, I guess, what happens behind the curtain around the products and the services that wind up coming and helping them do their jobs every single day. And so I'd like you to tell us more about your job as a validation scientist. What do you actually do, and what does like your average day look like?
So as a validation scientist, our job is to help manufacturers design a device that can be cleanable, disinfected, or sterilized. And usually that is a collaborative effort between the manufacturer and the third-party laboratory. So the users are not even in that realm. We are helping them design devices, as I said, for cleanability and sterilization. And as a validation scientist, we're also helping develop instructions for use that the users are using on the other end to ensure that with this design, the instructions we're putting together can be validated and can be reproducible if it's used in the field. So that's what a validation scientist does. But my day-to-day job is I oversee protocols that we write for so many different types of devices, whether it be orthopedic, dental, home care. We are seeing a lot of home care devices as well coming through. Yep. So we always have to think outside the box. We don't have an instruction for use that's set like we have for sterilization. Here are your parameters you have to use and you have to follow. If you don't fit into that box, guess what? You have to redesign. In our world, when it comes to cleanability and disinfection, we always have to put our thinking caps on and figure out how can we make this work? When we got keyboards, how do we clean and disinfect keyboards? Right? That was a challenge. UV disinfection. Does UV disinfection work? Will that work? If not, we have to use chemicals. What if they don't have certain types of chemicals in different countries? How do we optimize that? So we're always thinking, we're always on the roll of what is out there and what can we do better.
I really want to sit on this IFU piece, and I want to take two parts to this. Before I talk about the IFUs and how you help companies write them, I want to start with, you had just said something about how there really are no IFUs for what you do, right? So you help create these IFUs,
right?
But how do you kind of set those standards for yourselves? And I'm assuming that you're going to talk to me a little bit about ISO, and anybody out there who's gone through the DNV certification for their department, specifically around sterile processing certification, is probably going to be somewhat aware of ISO 13485, which is related to healthcare organizations, but I'm not sure that that totally fits into your world. So how do you do that process and create your own processes and then continue to optimize them and focused on quality control?
So like you said, there is the ISO 13485 for quality, right? We also have other ISO documents that give us instructions, ISO 17664. They give you instructions of what needs to be included in an instructions for use. We have AMI documents that we have to follow. We have ASTM documents that we use to simulate how we're going to perform these validations. And before we even go into instruction for use, there's a series of R&D studies that are performed to see if the instructions that have been written can give you the results that you need. So there are many standards that we have to follow internally just to make sure that we're compliant to and we're following regulations for that as well.
That is so interesting. So, I kind of want to walk through that process a little bit. When the IFUs come to you, I'm assuming that they're somewhat pre-written and you're validating them and or making recommendations for adjustments to the IFU. Is that correct? Do I have that right?
You have that absolutely correct. A lot of the times, these are 510K submissions. So they are using predicate devices to make a claim for their product, and usually are using IFUs of the competitors or the predicate device that they're using. And these IFUs are meant for other devices, but usually manufacturers do have an IFU outlined for their devices. And we follow those. And if we don't see that they work well, say a lot of time we have lumens and not sufficient volume is written in an IFU, flush the lumen. Well, how much are we flushing it with? That is what we're validating. You need to put 60 mils through the lumen using a 30 mil syringe, and you need to flush that twice. That is what we're validating. Now, does that language go into the IFU? Probably not. It's probably going to be general. Those kind of instructions you will see for gastrointestinal flexible endoscopes, but not generally for general instrumentation.
That's such a curious topic. You're pulling us into the IFU discussion.
You could just totally derail us from the point of this season.
Don't go there. But it does tie in to the chemical theme of this conversation, because obviously within all of these IFUs and all the testing that you're doing in cleaning and temporary processing, there's a chemical component likely. So when it comes to those chemicals, how do you approach that portion of the IFU and or that portion of kind of the validation testing?
So when it comes to chemicals, we do have different markets when we're evaluating the instructions for use. Any devices that will be marketed in Europe, we're using alkaline detergents for the validation. This is where we see Neodisher Mediclean Forte, Neodisher Z, detergents that have pH from 10 to 13. Anything US based will be enzymatic detergents. And that's what we use for those type of validations. So it is a bracket when you look at it. Neutral pH enzymatic detergents or alkaline. Now, what we use for the validations may not be what the users are using, right? The chemicals will be different. And we know that in the industry. What a third-party laboratory is using for the validation may not necessarily mean that the hospital down the road is using that same chemical for the validation. They're going to be different. So that's where they're grouped in these brackets. If it's a neutral pH detergent between 6 to 8, they should work efficiently. Now, what kind of enzymes are in these chemicals also make a difference. There are a variety of enzymes out there, and some have one enzyme, some have two, some have three. And this is where the complications start to happen when we talk about these type of chemicals and validations.
So you brought up that kind of specific chemical testing that's done, that's being validated. And I know there's probably some nuance here, right? So I want to ask this question kind of broadly, and then you can answer however you want. There's a challenge on the user side when we view IFUs and there's a specific brand that's listed in the IFU that says, soak this device in this brand for this amount of time. Is that generally the kind of thing that would come from a recommendation from the validation testing portion? Or is that stuff that kind of comes from that original IFU from the manufacturer that then is flowing, you're validating according to their IFU, and then it's just going on down the line to the user? Or is there some other way that those kind of brand-specific recommendations are getting in there?
That's a very good question. It is both. Sometimes we do get a detergent specifically specified by the manufacturer to use for the validation. And most of the time, they do not specify. So then we are going to use what we have in the lab that we use for validations. And that is specified in their final report, that your devices were validated with this detergent. And so usually they put that in the IFU.
And so, again, I don't want to put you on the spot if you can't answer this, but I know users are going to have this question. So if we see an IFU that has a brand name, are we able as users to use a comparative chemical, but outside of that specific brand that's listed in the IFU, or is that a question that someone else has to answer?
Not necessarily. I can answer that generally. We have to look at the MSDS of the chemical and see what the main ingredient of that chemical is. And we are currently looking at working together with an ASTM collaboration group to answer your very question, because that is something that we have been approached in the standard committees, and this question has been arised by many, and we don't know the answer. So we have created this group that's going to do a round-robin testing and see if that's even applicable, or do we need to do more to understand these different levels of detergents? Because now detergents are something that is being evaluated in the regulatory realm as well.
Yeah. I knew I wasn't the only one who had those questions, so I'm not surprised that it's coming up in those standard committees, as you mentioned as well, because it's just such a real life challenge when you're dealing with any kind of chemicals in IFU. So I appreciate that insight, and has great to hear though that there's progress being made, and that it's conversation not only on the user side but on the side of manufacturers and validation labs. So that's fantastic. Going back, to the specific testing of these, as you're validating an IFU for a device, are you actually validating that chemical itself, or the process, or both, or kind of what's that combination of things that's actually being tested in that whole scenario of device and chemical?
So when we're validating the device, the chemical plays a huge role in that. And so we are validating the chemical with the device and the instructions that we're using to clean this chemical. So it is both of them. And we are evaluating both as well.
So are there scenarios where a chemical manufacturer would reach out to you and say, hey, I want to know, does my detergent work? And so then you apply that to testing of various medical devices, because I feel like we've been primarily talking about medical device manufacturers coming and saying, hey, can you validate these IFUs, with various chemicals? Does it also happen the other way? And if so, how is that a different approach to what you do?
So we have had opportunity to work with detergent manufacturers. Usually we are working with coupons and not medical devices. So when we're evaluating those detergents, we are working with different types of coupons that are used to make medical devices, but the design features that the medical devices have, these coupons usually don't have. So,
right, like the hinges, the shadowing.
Right.
Okay.
Yeah. So we do work with them that let's make a PCD out of the coupons, right? Let's make it more challenging. So we'll drill some holes in there. We'll add a spring here, things like that. So we have had the opportunity to work with them. They've given us a broad range of different detergents, and they see the efficacy, how well the detergent cleans this PCD that they have created. So that has happened. Yes, they are actively working on that for sure.
And now, when you're validating surgical devices in particular, what kind of contamination, for lack of a better word, there's probably a more technical word than that, but what kind of contamination are you using in the device to test those IFUs and to validate the chemical uses? Do you use different types, or is it like one kind of standard medium that you're using?
Are you talking about test soils, Hank?
Yeah, that's the word. Thanks, Justin.
Yes, we do use a variety of test soils for the intended use of the device. So first thing we have to look at the device is, how is this device used? What type of patient soil or native soil will be on the product? And from that, we would design an artificial test soil, right? There is no one test soil that's used broadly across the board. We are looking for intended use of the product. And so orthopedic would be different, dental would be different, home use would be different. So looking at all those different scenarios, we develop test soils and we make test soils. And there's standards out there that give you a list of test soils that you can use on different types of products. With that, we pair that up with chemicals that have at least protein in it. So it has protease, which is an enzyme that attacks protein. And all test soils will have protein, because we say one of the test soils we commonly use is defibrinated blood soil. That's used for any devices that come in contact with blood. And that's used from sheep blood and egg yolks and hog mucin. And we grind that together, and that's what's used to contaminate the device. Now we're going to challenge that with a chemical, but we want to make sure that this chemical has at least protease in it. There are many different chemicals that may have amylase to get the carbohydrates, lipase to get the lipids, but we make sure that at least protease is one of them that's in the chemical. So it depends also on what type of device it is, what the intended debris will be on the device and the test soil and what type of chemical you should use for it as well. Because all different chemicals have different enzymes and they attack different parts of the molecules that the blood and the patient debris are made from.
So is there any kind of time factor in there? Because one of the things I know, about proteins, especially when you're doing a surgery and the individual is alive, is that once that, protein is on the device, the more time there is, and that protein then begins to denature, it kind of adheres to the instruments in many cases. So as you're going through that validation and even as you use the proteins, are there any kind of time considerations for how long it sits onto the device? And when you're mashing that up and making it, do you kind of test it before it denatures and after it denatures, or are we not at that level of testing yet?
We're not at that level of testing. However, when we're doing validations, we're doing worst case testing. That means the test soil is dried onto the device. So we're assuming that pre-treatment has not been performed and it's going straight to the central processing to clean. So when we contaminate, it is dried on test soil that we are now challenging to the IFU and the cleaning instructions.
Oh no, that's justification for no point of use cleaning.
Do not try this at home, ladies and gentlemen.
Do not share this with your OR counterparts. Just kidding.
You could do this because, yes, we have validated instructions, but let's not.
Yeah.
So this is an opportunity that we've kind of laid out here for you to bust a myth or two around misconceptions out there. So what are those kind of common misconceptions that you hear folks maybe reference out there during webinars or education, or maybe see articles about, not that anyone has any nefarious plans to kind of miseducate, right? But, from someone on the inside who's got that true experience, that scientific background, the microbiological expertise like you obviously do, how these things actually work and what the challenges are and what they're not. What would be those kind of like one or two things you're like, people are out there kind of confused about this, and now's the time that you can kind of set the record straight?
So one of the things that I often get questions about is, like we discussed earlier, the detergent you use for the validation, was that the same detergent that we are using here? And the question is no. And because manufacturers test for the world, they just don't test for United States. It's going to be different from city to city, to country. It's going to be everywhere. So what we validate is assumed that if you're in the enzymatic range, the enzyme should work the same, no matter where you are. And so that is one of the misconceptions, is that if you're in this group, all the detergents in the neutral pH enzymatic should be equivalent. But are they really equivalent?
That hits a theme that we just talked about on the last episode as well, which is how we got to the whole, how can departments verify that the detergent, enzymatic or otherwise, is doing, what it says it can do.
I wanted to ask a follow-up there. This goes back to maybe season 1. We had on Peggy Spitzer, who's one of our enzymatic experts for the Beyond Clean Expert series. And, so one of the bombs that she dropped is it's fairly easy out there to create an enzymatic and get into the market, because there's not a lot of clear definitions, from anyone on what qualifies, what the claims are, etc. And I've had this, crazy idea of one day coming out with a Beyond Clean detergent. So, from your perspective, if I were to build a new detergent out there, what does that chemical, what would make the best sterile processing detergent on the market that would check all those boxes that you're constantly testing against for medical devices?
Well, that is what we are working in our ASTM document. We're testing all the detergent manufacturers are part of this collaboration group, and we're testing their detergents.
So I need to get on the ball, is what you're saying.
Yes, yes, you need to join. Yes, you need to get into this testing group, and you will be amazed. I'm just going to say something that's very, very simple, but water alone is somewhat equivalent to many detergents that are out in the market right now.
Whoa.
And are comparable.
Justin, that's the thing. Beyond clean water. Let's do it.
Yeah, it doesn't even have to come from a spring.
Beautiful. So affordable.
Wait, so hold on. Hey, to go back to the last episode, we now know why that rumor about somebody drinking their own enzymatic detergent was floated, because it's really no different than water.
Just water. It was colored water. It was maybe apple juice.
That is, you know what, that really segues us really well to the last question, because honestly, I think that's kind of concerning. What you just said should be concerning to people, because if there is no chemical action that's actually happening when you're treating these devices, then it's really, as you said, no different than just running water over them. And then you're really relying on mechanical action for cleaning, and that mechanical action could be impingement or otherwise, but, you know, there is no enzymatic, or even the detergent itself, you know, if it's non-enzymatic, doesn't seem to necessarily be doing anything either. So with this in mind, I want to talk about trending as we close and wrap up here. Is it becoming like more difficult, or is it easier to develop these, you know, cleaning chemicals that are really going to work in sterile processing?
I think in the future, we will have a cleaning agent that will help the users use it for their purpose. And if we can harmonize with the testing lab and the users to show that what we're validating and what they're using is the same chemical, it does help. And I think that is where the industry is moving forward, is to ensure that we don't have 50 different detergents that we're actually using for cleaning these devices. And there is a few that are available and do a great job, because there are many out there that are equivalent to water, and they're being used. And like you said, our regulations around detergents used for that, there may be other regulations, but FDA is not, they only look at high-level disinfectants, right? And EPA looks at intermediate and low-level disinfectants. Who's looking at the detergents? Mm-hmm.
Yeah, that was the bomb that Peggy dropped on us, which we thought was so interesting, was the regulations really low. And the thing that really stuck out to me too, that you said that's an aha for me, is the way that the testing goes. There's certain proteins, you know, or protein-like substances, and, you know, including ones to mimic carbohydrates or fats, etc. But at the end of the day, they might be tested depending on if they're used in ortho or if they're used in eye, et cetera, et cetera. And when I look at all the moves around standardization, what I'm hearing is one size does not fit all. And so you could buy a detergent with that sort of fits the bill for one device, but then you find out that it doesn't fit the bill for another device if we even got to the point where we had something more advanced than water. So I would think that ASTM and all of that is also looking at, we really need a detergent that's going to be able to meet the needs of all the different types of procedures and environments that the instruments are going to be placed into. Is that also correct?
That is correct. But when you have detergents that try to meet every need, now you've created this major detergent that may clean everything, but leave things behind that can affect patient safety. So what are we doing? Yes. So we need to worry about that as well.
Yeah.
Those are the residual enzymes that are left behind that are very sticky, that did remove everything and did a fantastic job. But now we have to look at the patient safety side of things as well.
This is why scientists get paid so much money, because you'll have to figure out these challenges. This is why we encourage kids, you know, to grow up and get good at science, because it's not an easy solution, right? If it was, then it would already be solved. And
right, and these are the arguments we have all the time in our collaboration teams. It's like, okay, let's add this. Well, how if this much concentration is left over? Yeah, it's going to get rid of this. But how does that affect patient safety? Like, will the residuals affect anything? And we're like, yeah, it probably will. So now we have to go back to the drawing board and now redesign that chemical.
Well, one thing we know is water works the same on everything. So Alpa, You did a fantastic job.
Don't quote me on that.
No, that's my quote. That's my fun. Alpa, thanks so much for coming on. This was a very interesting interview, and I can tell you that 30 minutes went by super fast. So really enjoyed the conversation. Thank you for being so informative.
Thank you. Thank you for having me and giving me this opportunity to be part of Beyond Clean.
That was Alpa Patel, Principal Scientist at Nelson Laboratories, LLC, and Hank. A couple of great episodes in a row, but really a fantastic season 15 here on Beyond Clean. I know Alpa's done, some of the conferences with us, and I just love hearing from sort of behind the curtain as we use that Wizard of Oz analogy or reference there. But, really getting an understanding of the opportunities around chemistries and improvement. It's funny, in a way, that we had this conversation with Peggy so many years ago now, coming up into we're almost 5 years complete of the Beyond Clean podcast. And, really the same issues are there. It's nice to know that there's like ASTM, et cetera, that are working on improving that and maybe adding some regulation. But, I can't imagine washing my dishes with water, let alone surgical instruments that are then going to go back, inside of a patient. I just think that's really, really crazy. And clearly we're going to be doing an update at some point. It would be great once all of that is defined to hear what kind of progress has been made once they've kind of finished with that working group, Hank.
Yeah, well, you kind of went on the other end of the spectrum. You got upset and frustrated that some of these chemicals are like water. And I got excited because I was like, water. If they can put water in a bottle and sell it, or water in a carton, like a box. Have y'all seen that boxed water?
No, not yet.
Kind of crazy. So yeah, they're selling boxed water. Well, if that can be sold, then man, how about some Beyond Clean water out there that can magically clean the protein off.
Stay hydrated in your department while you clean your instruments.
Drink it and, use it in your manual cleaning sink, put a couple of bottles of Beyond Clean water for your automated washers. But, what's curious is obviously we do use water to clean in all these other ways, but we typically include active enzymes and detergents along with that water. So yes, water alone typically is not the objective. And to that, we agree. But terrific interview. I love it. As you said, getting that behind the scenes look. Just like our theme of Beyond the Tour, going into these manufacturer headquarters where most of our technicians will never get the opportunity to visit. Same thing with these interviews is we get to go behind the curtain, behind the scenes, and all these kind of chemical conversations and talk to people that for the most part, you're never going to get the opportunity to just sit down and have a 30-minute conversation with. So I freaking love this conversation and I love this podcast. What can I say, Justin?
So do I. And I love opportunity, like you said, but I'm not looking to sell water. I want to create the product that does verification of the detergents in the department. I think if we could sell that, then we'd really be on to something. But that's going to do it for this week's show. As a reminder, you can help support Beyond Clean by subscribing on Apple, Amazon, or Google Podcasts. You can also find us on Stitcher, iHeartRadio, Spotify, or search for Beyond Clean on your favorite podcast application. You can also access bonus content for certain episodes, but you got to download our smartphone app for iPhone and Android. We'd certainly appreciate a rating and a review while you're there because your feedback is important to the show. If you have any topics that you'd like us to cover on a future episode, all you got to do is send an email to info@beyondclean.net. And on behalf of Hank and myself, thank you for listening to this week's episode of Beyond Clean.